The overall goal of this procedure is to measure the amount of each phosphoinositide species in cells under various genetic and environmental conditions in order to understand phosphoinositide function and regulation.
Phosphoinositide lipids control cell functions like migration, replication, and membrane trafficking. This method identifies genetic and environmental conditions that impact phosphoinositides and increases our knowledge about their regulation and function. The main advantage of this technique is that it permits accurate, concurrent measurements of all phosphoinositides inside cells.
Though this method can provide insight into the regulation of phosphoinositides in yeast, it can also be applied to cultured mammalian cells. Individuals new to this method will struggle because of the many steps required for labeling, processing, and analysis. We recommend practicing without the radioactive label first.
To begin, grow a 20 milliliter liquid culture of the yeast strain SEY6210 in complete synthetic medium at 30 degrees Celsius with constant shaking to mid-log phase. Next, transfer a total of 10 to 14 OD of the yeast cells to a 12 milliliter round-bottom centrifuge tube and centrifuge at 800 times G for 5 minutes. Decant the medium and resuspend the pellet in 2 milliliters of inositol-free medium. Centrifuge the cells again, and then resuspend the pellet in 440 microliters of inositol-free medium. Incubate the cells at room temperature for 15 minutes. After the incubation, add 60 microliters of